All right, I'm gonna hit the other. Okay, supposedly we're recording now. Yeah, so I just dropped in the chat. Hi, everyone. This draft agenda and the updates are following. Oh, good. We got an agenda document. Look at that, Meredith. You're awesome. Thank you. So you don't have to do all those texts if you just open that one Google document. I think we have a guy on the agenda today, and I'd like to go and just go around the horn here with introductions. Any questions about the agenda or additions, deletions, complaints? Okay, so let's just do introductions first, and then updates other than those shown below, which is these. We have an NREL update. Yasmin, if you want to add to that from Alaska, I don't know if you're doing anything up there. So anyhow, let's just do updates other than those shown. So I'm going to do this in reverse alphabetical order. Let me see here. Zender, but no. I think it's going to have to be you anyhow, Simone. Simone Savallo, please.
Hi. Yeah. I'm
And
Simone
then Kristen.
Savallo. Say it again.
Then Kristen.
Oh, then Kristen. Okay, great. Yeah, so I'm Simone Savallo. I'm the deputy director at Zender Environmental. We're a nonprofit out of Anchorage, and I haven't been on the last couple of meetings, but yeah, I was curious to see how the NATO funding stuff is going. I've been keeping up with that. And I can't actually stay on long today, but I wanted to pop in anyways to say hi. It's great to see everyone.
Now it looks like it's light outside your window where Meredith is saying it's just starting to get light.
Yeah. Yeah. I'm actually I'm in Northern California.
Okay. Go ahead, Kristen.
Hi, this is Kristen Reardon. I'm at the Denali Commission. So we're a small independent federal agency. We're operational through the shutdown. So we're here, and we're based in Anchorage, but we serve the whole state, rural development throughout the state.
Cool. And then Yasmin, followed by Margaret, and then Jane.
Hi, I'm Jasmine. PhD student here at the University of Kansas, and also have some affiliations with Zender before I came. But just a little update for me. I have met with Tom Curtis, and so we made a connection in terms of getting my PhD work, the experimental design kind of locked in. So that was good. That happened a couple weeks ago. That's kind of still in the works, but it's exciting that we've made the connection and confirm that we can collaborate together. So
who's Tom Curtis? Refresh our memory.
Tom Curtis, I feel like I wouldn't be able to give him like a great introduction just because it's still new in terms of the relationship, but he actually came to the conference in Fairbanks. So he's a researcher. I can't say from what university at the top of my head, but he's really well known within like my microbial community, specifically psychrophilic methanogens, which is great. And just like inoculating biomass, what I'm wanting to do from the Arctic, and then putting that in an anaerobic digester. And so he's done a lot of research in that area, which is great.
This is the guy who collects lake sediments.
Yeah. Mm-hmm. Okay. And I think that he does a lot more things than that, but right. Yeah. He used to come to some of these meetings, but I think that he's gotten busy, but he had mentioned getting back onto one here soon. So maybe we'll see him around.
Cool. All right. Thank you.
Are you doing your PhD with Tom then? Are you doing your PhD in Newcastle?
No, actually, I'm here at the University of Kansas, but because of my advisor, Dr. Belinda Sturm, they know each other within the area of wastewater and water. And so I'm like the middle person. I made the connection, and then he said that he was good to go in terms of collaborating. So he might end up actually being on my committee in a way. So yeah, it'll be good. I'm not there with him, but we've definitely solidified a collaboration, which was great and super exciting.
Good. So that leaves Margaret and then Jane. No, Margaret and then Nate and then Jane.
I'm Margaret McCauley. I'm with the United States Environmental Protection Agency. The United States Environmental Protection Agency has a tribal trust responsibility and many requests for poo solutions for communities where piped solutions are not solutions, but just liabilities. And so we're trying to figure out what might work. I'm so delighted to have all of you along on this journey with me.
And Margaret is a co-founder, let's just say that. Or co-instigator or conspirator or something.
And I'm based in Seattle, where I'm in a regional office that we cover 4 states, including Alaska. So who was after me? Nate was after me.
That's right.
Hello, so I'm Nate Janiga. I'm with Corollo Engineers. I'm focused in wastewater and water reuse, but I'm a consultant these days. I used to build membrane bioreactors for a different company, Kubota, and but I've been pulled into this because I've been working for a long time on the Mount Everest biogas project, which is very cold and remote. And very hopeful biogas design that is like wildly stalled right now. Right now I'm trying to get our website back online. Anyhow, I'm very personally invested and interested in small scale anaerobic digestion. This is an amazing project. So yeah, I've been here for a while. I'm in industry, and Embry Bronstead isn't here, but I'm going to steal her tagline of anaerobic digester proselytizer. Was that her? Anyway, yeah. I'm a nerd. I like this stuff. Anyway.
I'm Jane Fowler. I'm an assistant professor at Simon Fraser University in the Vancouver, Canada area. I'm also the NATO country project director for the NATO project. And so Simon Fraser University and myself are kind of administering that project, and I can give you guys an update on that more later when we get to that agenda item. Yeah, I've been coming to these meetings for, I think, like 5 years now. I was actually dragged into this by Tom Curtis, who is, a super experienced guy in studying psychrophiles and working towards making anaerobic digestion work under cool temperatures. So, yeah.
Thank you. Okay, Anne E. Corrigan, you're up. Welcome. You can unmute and you can tell us, give us your introduction and any update apart from the NREL update, which you can give us in a few minutes. Any other updates besides the one that you emailed me that we'll talk about in a minute? Or like 10 minutes or 20?
I don't think I have any updates, but I'm Annie Corrigan, and I think everyone I've talked to before, but in Fairbanks, and we have, I'm not supposed to give my Fairbanks updates. We have the pilot experiment going on for right now in hibernation around anaerobic digestion at a small scale, seasonally operating. Other than that, I work on non-AD related technical assistance energy projects in Alaska at the National Renewable Energy Lab. Yeah.
And for those of you that just joined, and Annie, as usual, I'm having a little difficulty hearing you. It always seems like your connection is a quiet one, but maybe you're just a low-key person. So there's that. And I just dropped into the chat for those of you who came a little bit later, the agenda as a Google document. Thank you, Meredith. But basically, we're going to start off with these introductions we just did. Oh, I'm Alex Topillas, Vermont Agency of Agriculture, coming at this because, garbage into gold is always interesting to me, whatever form it takes. This is really just a side interest of mine. My core duty is energy and climate opportunities to improve those for Vermont farmers, and anaerobic digestion checks both boxes. However, I'm interested in this group because I'm interested in something small and robust for the smaller farms that we have here, 50 head, 200 head. of dairy cattle. And we've got sheep and goat dairies too, and beverage and creamery waste. So all of these things are happening often on a small scale. That's why I'm interested in this technology. Not for sanitation, not my thing, but there we have it. Any other questions, alerts, anything before we get into Guy Roberts?
Meredith, intro. I don't think we got.
I'm gonna have to jump off in a little bit, but I really wanted to join, so that's my
okay.
only thing.
Meredith is our awesome fixer. She's like in that movie. Never mind. She's does all this wrangling of us in our meetings and getting our agendas out, and it's all part of IRPIC. Is that Meredith? What do you want to add to that?
Just that I'm based in Alaska, and so what you guys are talking about is is interesting to me and feels meaningful. So keep it up. And I will be jumping off in 10 minutes because I've got another meeting, unfortunately, that's always scheduled at 9 a.m. Alaska time on Wednesdays.
Okay, well, we expect you to stay for at least 10 and a half more minutes. And you're studying something, aren't you?
Yep. Yeah, I am in the process of becoming a mental health counselor here in Anchorage, about to graduate from UAF in December.
I can tell by looking at you, you're going to be great.
Thank you.
Yeah, my mother was a social worker, so I certainly value that stuff. Okay, Guy, I'm going to allow you to introduce yourself. I'll just say that I met you through when I came here 13, 14 years ago. You were prototyping and building commercial scale barrel type digesters for small applications, food waste, manure, etc. As a company called Avatar Energy. So yeah, you have a microbiology background, if I remember right, and you're now sort of managing facilities at the UVM, University of Vermont, including labs. And Guy also worked for like 3 or 4 years here at the Agency of Agriculture when we were building a whole new lab and ran that lab.
That's right. And then my daughter needed to get to school, and I had to get back to find it. As they moved me across the state, I had to move back towards closer to her high school so I could get her to school. But let me thank you, Alex, very much, and thank you all for letting me join you. And I'm looking forward to sitting in on these meetings going forward, if you'll have me. Let me share my screen, and I'm just going to share this window, and hopefully this is not presentation mode, but hopefully it works out. Let me know if any of you have difficulty seeing it. So this is kind of a splash slide. I'm going to go over my own background a little bit, and then why I got into small scale anaerobic digesters, and just a brief splash on the commercial scale work I was doing at Avatar. But then I want to talk more about some of the challenges that I've found working on very small scale digesters. I've been working on a digester in my garage, and that I kind of treat like a village. And we'll talk about some of the two particular things. One is mixing, and another is some of the, well, we'll get there. Alex, you had a question?
I did. I'm sorry. I just realized something logistical. Several of you came and may have to leave. For those of you with an update, for example, we need to like touch on these while everyone's here, ideally. The NREL update, I would love to hear from Jane, and the NREL update, I said NATO update from Jane, NREL update from Anne. Can you stick around until Guy is done in 30 minutes, or do you need to offload now? Or whoever was going to do those 2 items? Or is there anything else burning somebody needs to share about those 2 items or the IWA stuff before they leave?
I can stay for half an hour.
Okay.
All good?
Okay, great. Thank you.
Okay.
Sorry, I just forgot that like we have people coming and going. Thank you.
No worries. No worries. Understand. So back to this, super. And then, so I'm going to show you some, I think Alex has shared some photos, but I'll go through a little bit more about how this small scale digester, appliance scale digester that I've been working on more or less as a hobbyist as works out. And I've got to figure out how to change. So I started out, went to college in Western Washington, in Bellingham, Washington, studying at Huxley College of Environmental Studies, and worked with a faculty member on human settlement design, more or less. Came away with a major called Human Ecology, Environmental Health, and worked on how to, my thesis project was how to dispose of waste up at on Mount Baker, at a climbing hut up on Mount Baker, where fecal coliform counts were starting to appear positive in the soils around the cabin. And I got to spend a lot of time up there, which was fun. I went on to study parasitology, and parasitology is kind of an interesting blend of microbiology, ecology, environmental studies, pathology, and it ended up being many years chained to my workbench in a cold room, a little bit like working in the Arctic maybe, but did a lot of separations of RNA protein complexes to study RNA processing in the parasite Trypanosoma brucei. Did a couple postdocs, more work in cold room, working with PCR as it came on the line, and pretty fun. But then a friend of mine invited me to join a startup company, Montclair Group, which was an incubator, wanted to start up several companies, and I ended up being placed as the chief scientific officer for an antibiotic discovery platform looking at tRNA processing, editing as a drug target in bacteria. And due to health crisis, I moved to the East Coast with my ex-wife, and decided to land in Vermont rather than New Jersey, which I think was overall a pretty good decision. And started doing some work on alternative energy, kind of small scale alcohol, waste to alcohol, but then landed on anaerobic digesters. And I'll tell you a little bit more about that. And then, as Alex mentioned, I worked briefly for the state of Vermont as a lab director. And now I'm the facilities director in the College of Agriculture and Life Sciences in the Dean's office, helping all researchers get the resources they need. So what triggered my interest in anaerobic digestion? Right around when I started thinking about moving to the East Coast, the Pew Oceans Commission released their report. And it was kind of startling to find out how many major rivers had dead zones at the mouth of them, and that it was due largely to farm nutrients that were escaping downstream, runoff and into the rivers. And it seemed like a win-win situation to be able to actually figure out how those farmers could retain those nutrients. And anaerobic digestion seemed like one of the problems with farm manure is that it's got nitrogen and phosphorus, but in kind of the wrong proportion for plant uptake. And so you end up loading your fields with phosphorus. And typically they spread manure at the time of year when it's right before the snow falls. And so spring runoff and whatnot invariably causes a lot of runoff into the surface waters. So looking at anaerobic digestion, it seemed that there was a way to separate these nutrients into a liquid fraction and a solids fraction, and nitrogen enriched in the liquid fraction could be spread on fields during the growing season when it's needed, and then the phosphorus typically adheres to fiber in what we've seen. And so you recover much of it as the fiber when you press the waste the effluent out of a digester. You can use that fiber as an animal bedding and kind of recirculate it in-house, so to speak, and keeping it and apply it as needed. You could, one of the things that was very interesting to me was potentially exporting it from the farm to vegetable growers, which have a higher demand for phosphorus than dairy farms do. So as I landed, I actually touched base with my previous PhD advisor, and she said, we have an investor who's interested in starting a company looking at water management. And what are you doing out there? And it ended up creating Avatar Energy, where I was working on plug flow digesters. The plug flow digesters were developed in the US, working on basically animal manures, high solids materials in the 12 to 15% solids range. And it typically digests down into something much more liquidy that can be spread or separated. But a lot of those were built as permanent installations with a limited lifetime. Concrete starts to degrade within the environment of a anaerobic digester. So I started looking for a way to make a digester a movable asset, something that could actually be sold if a farmer decided to move on and sell their farm, as many farmers were at the time. So that wouldn't, and then something that would be cost effective, but with a long enough lifetime that you'd have plenty of time to achieve the return on investment. And the way that company went is a story for another time. There's some catharsis involved, but we don't need to get into that here. So I left that company in 2014 and did some work on my own briefly. Had a microscope and did travel, but did a little consulting work before I landed at the state. But I wanted to talk now about some of the challenges that we ran into in small scale anaerobic digestion. And so first, some photomicrographs that we took of the bugs growing in our digesters, and we were purposefully inoculating our digesters with wastewater treatment facility effluent. And what we found was growing in those were primarily Methanosarcina, this kind of packet-shaped bug, archaea. And it turns out to be extremely robust. It can grow in mesophilic environments. It can grow in thermophilic environments. It now has proven to be able to grow down to about 80 degrees and to still be productive, 80 Fahrenheit. But one of the things we noticed was that it typically grew in association with some kind of fibrous particle. It was almost always attached to something. And that became a really important factor, because as we developed our digesters, initially we were working with systems that we thought were mixed, but weren't very well mixed, it turned out. And so I want to just briefly talk about the importance of mixing. And I'll tell you about an experiment that we did that really underscored the importance. It turns out that in an unmixed digester, very quickly you'll form a crust. And I think it's largely because as these organisms produce biogas, for some period of time, the bubble is attached to the organism and it floats along with the fiber, and it takes up to the surface of whatever slurry it's growing in. And over time, it creates a crust. And that crust is, it can form within a matter of weeks, days to weeks, actually. And I've always been kind of wanted to know, where do the bugs live inside the digester? Well, we added vegetable oil to one of our digesters, knowing that it can really kick a digester. Typically, you don't want to add it at more than 2% because you can really shock the system. And it killed our digesters. We were using just small batch digesters at the time. And as we looked at it, we realized the oil had actually created a layer between the crust and the liquid beneath it. And so it was cutting off the crust from the nutrient solution below it. And it was starving our methanogens. So we realized that, wow, we really need to be mixing these. We have to be very careful when we add oil for even a new reason, but we really should be breaking up this crust and reintroducing it into the solution below. So that really drove home the point that mixing is important in some capacity. And so when I see these tubular digesters that are made out of plastic or rubberized tubes, I was thinking about my experience at Avatar, where over time we got those just filled with kind of bread loaf texture crusts, and it just filled and filled and filled until there was very little water running underneath it, and that crust pretty much occupied the entire thing. cleaning that out is an incredible effort. Takes an incredible amount of effort. You're carving it away if you have to open it up and go inside to clean it. So these systems have a fairly limited lifetime until the crust overwhelms the system, unless you can mix it. So to be able to introduce some kind of mixing, whether it's stepping on it, or whether it's introducing some kind of a paddle, then if you can do that, then you can avoid this eventual filling of the vessel with crust. The second issue that we ran into was the fact that we're trying to optimize a mixed system. We're trying to optimize multiple variables and mathematically, we know that's not really possible. You can optimize one variable at a time. And yet we have at least 3 variables inside of a digester. When you feed it your organic waste, it gets processed by a group of organisms that we call heterotrophic bacteria. And they break it down into small intermediates, alcohols, organic acids, etc., and then sugars. And then the acetogens have their way with that to create acetic acid. And this is only one group, and it's only one pathway towards methane, but it's a pretty important pathway from what we've seen. And these tend to like a fairly acidic environment, pH 5 to 7. Then you come to the methanogens who eat the metharsarcina in particular. They can eat this vinegar, but they don't like acidic conditions. They like neutral, pH neutral or slightly higher conditions. So to get in one vessel conditions that meet all of these is a little bit difficult. So how do you mix this digester and not create a homogeneous set of conditions that's not optimal for any of your bugs? So pH, redox, potential small molecule concentrations. How do you achieve a blending that doesn't become a compromise for all these organisms? And when you continuously feed, you're also running into a problem where you have some coming in and some coming out right away. So we started looking at tubular digesters primarily because they became a fan of tubular digesters, primarily because as you mix these, as long as you're not pushing the material forward as you mix, but just turning it over, you're actually just maintaining environments that allow for your heterotrophs, your acetogens, and the methanogens to thrive within their own environments. So you can have different conditions setting itself up within your digester. And if you measure pH along this, which we've done a little bit of, but our digesters weren't bristling enough with ports and stuff for us to actually really do the work we wanted to do. You would see there is some pH differences, low to medium to a more neutral pH from the beginning to end. I've always wanted to look and see whether you also see variations within the population of methanogens where through gene expression variations, they actually express fewer receptors for acetic acid, or maybe less affinity in those receptors versus being more miserly and more tightly bonding to acetic acid towards the end. So I imagine there's probably just within this distance in the digester, there's also variations in gene expression within the methanogens. And so this is somewhat speculative, but depending on the type of vessel you're digesting in, you're varying the completeness of digestion. So I'm estimating, and I've never measured this, but again, as you put material into a complete mixed digester, some of it comes right back out. So you do have some underdigested material. Some of it is in here just for the 18 days or whatever you'd like to have it, and it comes out at that point. So you get some that's reached your full retention time, but then some will just stay in there for too long and you'll have it over digested. And that might not be a bad thing because that tends to re-inoculate your digester. So there's benefits to that as well. But in a plug flow digester, you should have very little underdigested unless you're really pumping material in here and it's creating a plume that's going from one end to the other. if you're adequately mixing this, you should be able to have material moving through in true plug flow fashion. And so you'd see a higher level of material that's reached your full retention time. And invariably, you'll have some that's collecting along the sides that's become over digested. So this mixing type is actually kind of important. And so I want to talk about the digester that I've designed and the mixing system. It's a tubular digester. And in this pencil drawing, you can see it's basically a 1 foot diameter PVC pipe, 4 feet long, with Vanstone flanges on the end, so I can attach plates to the end. And I've attached bearings in the end plates, end walls, and there's a stainless steel rod that runs down the length of it. And on that rod, I've mounted 4 basically pulleys, and I've cut 3 of the spokes out, so they're 3 spoked pulleys, and then I mounted these scrapers on them that as you move in one direction, it scrapes the outside of the digester. In the other direction, it just gently mixes the digester contents. And I might, if it worked, have a video here that we can see how that works. My assistant. So you see how that in this direction, it's pushing the plates out so that it scrapes the digester outside. And in the opposite direction, they just gently flow. They don't scrape as hard. They just kind of reach kind of a feathered position, so to speak.
What's that material?
That was just water. That was just a test.
The flaps, scraper.
Oh, the flaps. That's it's a low density polyethylene or polyester. So LDPE, that you can buy in sheet format. So this is the digester. I didn't put a slide in here to kind of back out and show you the whole thing, but we'll get to that, I guess. So there's feed tanks up above. There's a tube located behind here that is the digester itself. And then this is a mechanism I mounted on the front of the digester so that I can have a pipe, so I can take from the feed tanks up above, pump them through and into the front end of the digester. And at the same time, if I want to, I can pump out of the digester and out into a waste tank. So I can pump in and out at the same time, which helps maintain the volume. I can also close one or the other and pump out or pump more in so that I can balance it. And because the ends are Lexan, I can more or less see what the level is inside. It'd certainly be nice to have a better level sensor. And I was using this, I had kind of, well, from my chromatography days, I spent a lot of time in that cold room sitting next to running peristaltic pumps. And I guess I became fond of them. So I built one to basically to be able to push 2 channels. So both the inlet and the outlet material coming through a different tube at the same time. And so you can kind of see, so this is just old ink rollers that I've kind of pressed out and replaced with metal axles and mounted in this Lexan end plates. And so these roll very nicely. And what you can see in this photo somewhat is a pinch of material that is going to be captured and pushed on through and pushed up so that it displaces material from the inlet side to the outlet side. And you can adjust it. It has springs that I can adjust the tension on, so I can pinch it, but not too hard. And it moves very nicely without allowing material to flow backwards. So one of the other things that I've realized, so here's a better shot of the whole digester with the digester itself sitting horizontally back here, and the feed tanks above the digester, the piping coming down into the peristaltic pump, the effluent end going into waste, the inlet end going into the front end of the digester. And after couple years, I realized that it's not that fun to take a digester apart. I was feeding this pretty much food waste from my kitchen. It's not that fun. So it's turned out to be convenient. I didn't want to turn this feed tank into its own digester by inoculating through it. So I just recently installed an additional tank that I can add manure to or inoculum from a municipal waste treatment plant to re-inoculate, because periodically with food waste, it's always an experiment to see whether this food is going to be received well by the digester. And you try and accumulate enough of it that it all kind of averages out. I can tell you about an experiment that we did with breakfast, lunches, and dinners. Bottom line is, it turns out that it likes to eat all three at the same time, not just breakfast, lunch, or dinner, because they kind of create a better mix, a better blend. And so if you do acidify it, you can get milk of magnesia in fairly large jugs. And a few of those in your feed tank can actually really help resuscitate a sour digester, but it often requires re-inoculating as well. So this is an inoculation tank that doesn't get feed waste. I can wash it out independently and keep this tank more or less pristine, uninoculated with food waste. It's far from pristine, I will tell you. And so the biogas storage, you can store the biogas just inside the digester, but you're then kind of limited to what you can produce at any given time or the rate that it's producing it. So I use the digester to push biogas into a tank that's filled with liquid. And then as it fills this tank, it pushes the liquid up into an upper chamber. And this is open to the atmosphere. It's not pressurized in any way. But the difference in height is directly related to the pressurization of the digester. So it's free to back pressure through this inlet tube. So the entire digester is pressurized by the distance. So if this is at 2.3 feet equals to 1 psi, this is about 4 feet. So it's just under 2 psi of pressure inside the digester. And it does just fine, because we know methanogens can grow at the bottom of swamps and things like that under much deeper water columns than this. So they can grow very well under pressure. So this does not impede growth at all. And then I can take off of this line to fuel a biogas burner, which is shown here. And that's what I've got for you. I have a couple of slides I could show how I've tried to envision how this would work in a restaurant setting, but I'd be happy to take any kind of questions you might have. Oh, Alex, I think you're muted.
Who else has questions besides me? Go ahead, raise your hand or just chime in.
I had a question.
Yeah.
Methane is such a escape artist. It's a really small molecule that likes to be in gas phase. Did you find any issues like with your bearings? Like I'm sure you have that waterproof. Yeah.
Yeah, the food grade bearings, they can be, in fact, actually, I noticed I'm smelling something right now. So I do need to go back in and grease them. The really important thing is to keep the level of liquid over the bearings, so above. So if I can keep the digester about 2/3 full, very little escapes in that regard.
Yeah. Okay,
but you're
awesome.
right. It loves to get out if it can.
I love the setup. I love the peristaltic pumps, and I really appreciate your hand-drawn sketches in ortho. Anyway, that's great.
Not great at SketchUp or CAD, unfortunately.
I guess I'll ask my question. Anybody else before? I'm not seeing all of our participants here. Let's see. Oh, I guess I am. So 2 quick questions. One, what is that material? I'll put a picture of it that you use to make your own peristaltic pump. What's that hose?
That is actually it is a PVC hose, but it's I did go, that's a great question because I did see several types of hose delaminate. So that one was one that I found. It's a lay flat hose. And I'd have to go and check the brand, but it wasn't a particularly expensive hose. It was available online though.
I'll put a picture in the chat, and maybe people know what that is. The other question I had is, where do you pull samples? Like, if you want to check things, like how are you doing that?
At the far end, I have actually at the far end, I have an outlet that goes straight out. So I put a T at both the front and the back, so I can actually draw from either the front or the back. And I can draw, but I don't have ports because of the scraping. I don't have ports going into the digester itself because it would block those, which would be a really nice thing to have. It means I have to put all any kind of, if I wanted to check, if I wanted to install thermocouples and things like that. So currently so this is heated. I didn't mention that. There is a heat pad, 2 heat pads from a pet store for heating aquariums, terrariums underneath it. And they're powered by a little unit that has thermostat in it. Those failed recently. So my digester is quite cold right now. It's in the 60s and it's not producing any gas. But I've just realized they have much nicer heating units for under carpet and under tile at the tile store. So I'm going to be making a trip over there to purchase one of their little heating units. They come in modular sections, all electrical.
So what
I'm
temperature
curious.
do you normally run it at? Sorry, Annie.
My garage is not heated, but it doesn't, so in the winter months, the garage itself gets down to 50s. So I can usually get my digester up into the 90s. In the summer months, it can get, it gets into the low. It spent most of last winter in the 80s, high 80s, and it made gas continuously through that period, through that time. In the summer months, it runs a little better in the high 90s, low hundreds. So I'm running at mesophilic. I'd love to get it up to thermophilic if I could. I think it's built with materials that could handle it. And what we saw when we ran one of our commercial digesters at thermophilic temperatures, we were able to feed it at twice the rate and get more than twice the biogas. And it turns out you don't have to change the organisms. These guys just adapt. They bump themselves up, or they can transition.
And just question, I like this idea of this inoculation tank, and especially for like the digesters that we're going to be designing.
Yeah.
It's a really good idea because we're probably going to have to re-inoculate like after the digesters have been frozen. But why did you have to separate the inoculation tank from the feed tank?
We didn't have an inoculation tank on one of our commercial digesters, and we very quickly but we had a mixing tank, a premix, and it was a preheat tank and premix tank. We very quickly turned it into a digester, and which becomes dangerous when you're sticking your head in there to check on your mixing system and whatnot. So the digester is vented. I've got it vented to the outside. So there's very little, it's very tight, but this feed tank is not tight. And so if this started producing methane, it would make this system much more difficult to have anywhere near my house, for instance, or in a restaurant or wherever. This inoculation tank, it's got manure. I can run it empty and then clean it out. And and then this becomes really important here because I can close a valve so that here and this so I can flush this out all the way. Close this valve so I'm not pulling anything out of the digester, and just flush this tank without doing anything to the digester itself. So a big part of the modification was this kind of dual valves here.
Yeah. How often do you like inoculate? And then how did you see the performance change when you incorporated this inoculation tank in terms of it getting better or producing more gas?
Yeah, well, the reason I did it was forgetting what I fed it. Something greasy, and it died pretty quickly, and the biogas production dropped off very quickly. So I gave it 2 liters of milk of magnesia, and then and that didn't. It slowly started to come back on. So there were some survivors in there, but it takes quite a while for them to grow back. Almost everything about running a digester to me is a population dynamics challenge. Feeding it, letting the mouths develop, letting the population get big enough that it can consume the acids as they're produced. And it just wasn't really recovering as quickly. So then I went and I work at the farm, or the university has a dairy farm. So I took a 5-gallon bucket of manure and put it into that inoculation tank. And within 2 days, it was cranking back along. So yeah, dairy manure is kind of the gold standard. The municipal waste treatment stuff is great too, because it's just we have a thermophilic digester at our local municipal waste treatment system, and they're very generous with their effluent. But it does take a little bit of, you have to contact them and let them know. Whereas picking up a couple shovels of, the cows don't mind if you take their poop. They give you permission pretty readily.
Have you ever experimented with like dog poop?
Yeah. Dog poop, dog food, pig poop, you name it. We did a lot. We had a whole series of batch digesters that we ran all these things in, comparing it to kind of our gold standard, which was dairy manure. It digests. It's all a matter of inoculation. The dog food didn't digest it out. It fermented before we could digest it. It started fermenting. Pig manure was a surprise to us because it's can be difficult to digest pig manure, but it digested really nicely. And I think one of the things that is important to recognize, in one of the reasons dairy manure is so robust as a feedstock, is it has so much fiber, and that fiber effectively buffers the digester. And so when you're working in low percentage feedstocks, for instance, in wastewater treatment, it's pretty standard to have to make regular adjustments of alkalinity and pH. And so you're doing that chemically with fairly expensive, sometimes additives, and in fairly large amounts. Whereas if you have fiber to feed it, even if it's coming from old lettuce or something like that, you're going to benefit your digester by buffering it more effectively. But you then you want to make sure that the reason I'm getting away with 1.5-inch pipe here is because I also put all my material through a big wearing blender. It's got to be the consistency of a smoothie, basically. But I feed it at about 20% solids. Food waste is remarkable. You can feed it really high and it still moves as a slurry. And then it digests down to close to 4%. So it comes out very, very low percent solids, which is great for pouring on your tomatoes or fruit trees or whatever.
You go from 20 to what?
About 4.
Coming up on 1:30. You infatuated everybody enough for Nate to stick around this whole time.
Wow, thank you.
It's a great presentation. Thank you so much. I gotta jump off. Thanks, folks.
Bye, Nate. Yep.
Cheers.
All right. So let's get to these other updates. Go
I
ahead.
know
Any more?
I'm going to
Yeah.
interrupt because I know
Yeah,
I'm
yeah.
one of the updates, but I wanted to ask, Guy, I think you had mentioned doubling as your biogas output depending on the temperature and season. Are you measuring how much biogas you've been producing and how much that varies with whatever you're feeding it?
Yes. And let's see if I can get back to, oops, actually, basically I can measure, I have a scale that I measure the height of the gas as it's being produced just by the amount of liquid that's being displaced. At this point, and we used to, when we were working with a thermophilic digester, we were running that gas through a generator. So we were monitoring the production rate pretty quickly or pretty effectively. And it was remarkable how much it really depended on being able to ramp up your heat input to get it up to that temperature and to maintain, to have great insulation so you don't have to keep injecting all that heat. But it was remarkable how rapidly it improved. It's stabilized. Running at a thermophilic temperature was both more stable as well as more productive. So if you can get up to that temperature, it's a much easier beast to manage. If that helps.
I just observed that this idea of zoning things, which is kind of inherent in a horizontal barrel, is pretty interesting. And I mean, you have such a small barrel, it's hard to imagine that really distinct communities exist. And yet at the same time, if things are going slow enough and you don't stir the crap out of it, then I suppose you may have different communities, just like there's, and so I'll just observe, like when we talk about thermophilic is great, we're in a situation for all of these sites where it's really a matter of, thermal management is huge. And you can't, just run crazy amounts of electricity into a heat blanket and, and get any energy. So I've been thinking like digestion systems that have a psychrophilic time and then, move into an actual mesophilic or thermophilic, smaller weatherized situation. And to me, what you're saying is, but you'd be concerned about making that transition in biology too quickly. Not only is there a transition through the stages, but there's just transition, which of these stages works best at what temperature. But my scheme is, spend very little money on psychrophilic retention capacity, volume, and then spend more money on the mesophilic, but enough so that you don't shock the microbes that are moving from psychrophilic to mesophilic, or for this matter, thermophilic.
Yeah, you're kind of talking, well, so multiple tank digesters is one way to approach that, so that you have, for instance, at VTC down up over in Randolph, they had a large pre-digestion tank and a digester, and whether they were scaled correctly, we'll never know. I don't think that's online anymore, but it is, and that is also one of the challenges. How do you scale a multi-tank digester?
And I think the way they did it was about 3% for the hydrolysis tank and 3 days rather, and 25 or 30 days on the main tank.
Right.
Because then both tanks at the same temperature, I think.
Right. And so if you had multiple mixed tanks that were at different retention times, you would effectively be pushing, you could have a tank for your heterotrophs. You could actually monitor it to see I'm not showing any of the data where it shows gas production, but you see spikes in gas. We used a very sophisticated device called a cigarette lighter to monitor our biogas quality. So as it came out of our batch digesters, we'd light the gas. Initially, it wasn't sustainably flammable. In fact, initially it wasn't flammable. That was just a big CO2 spike coming off, and that was a huge spike. But then it would go into a partially sustainable flame. If you kept the lighter going, it would burn, and that was probably hydrogen and hydrogen sulfide. And then it started being sustainably flammable, so you could tell that was the methane coming off that.
That's why they call them methanogens, dude.
Yeah, and so you could actually monitor the gas production from your tanks. If you had multiple tanks in series, you could be monitoring them to make sure that your retention time is just what it needs to be. It doesn't
All right,
need.
I've taken enough time with my nerding
Yeah.
schemes. So anyhow, let's.
Yeah.
It's 1:32. Let's move on. This has been great. Any last minute, tell guys full of shit or full of food waste or
Yeah.
something, as we all are, by the way. We're all full of that stuff. So anyhow, anything else on this before we? Okay.
Thanks again.
Yeah. Thank you. All right. It doesn't matter what order we do this in. Anne E. Corrigan.
Yeah, I'll be really quick. It's cold here. And the power system we disconnected last week, we brought batteries inside, so nothing's being heated out there. And I'll be making the last set of what did the season look like in terms of temperature variance across the tanks, doing some of that visualization in the next month. So we are firmly in hibernation over here, theoretically, if it were digesting.
So Guy, her assistant, was the guy that I brought to your prototype to show.
Oh, okay. Yeah. Yeah, that was
Wilder.
really Wilder. Yeah, really a pleasure to have him come visit. That was fun.
Yeah,
I don't know that
reported
we got him to
great
be
trip.
a acolyte, but there's still time to maybe proselytize.
Yeah. Okay. Well, send him back out.
Yep.
We
Anything more on NREL system? We'll look forward to your stuff. We love data, so whenever you have it, that's cool.
Okay, great. Yeah, if people have questions, I'm open to it, but I think I talked a little bit last time. We looked at some updates, but really just waiting for what the last month has shown. We can look at in future meetings. I'll just give you a fun FYI. Separate thing is that if you all were aware, the waste to energy project with the Alaska Energy Authority.
Oh yeah, Sean.
Around fish waste. Yeah, so that's actually all of the fish waste is currently being shipped to NREL and Golden to get feedstock composition analysis. So that's still moving.
Huh, cool. Stinky containers of fish waste are on their way. How do is this all refrigerated, or what do they do?
Yeah, they're sending it frozen. And the directions are to make sure it's really sealed, or our receiving will not be happy if it's visibly leaking.
Right.
But I think that's all there is to it.
And I told Sean via LinkedIn that Chomp Energy is doing a fish waste pilot characterization or something like that. So he was pretty interested in that, was going to follow up. And I guess you also, this is a good segue. You said in your update, I also have a meeting to learn more about NREL contract options and catch up with Jane in case any of the NATO award would come this way. So NATO, Jane, there you are. But what's her name? That was actually going to give the update, Sabalo? Or can you tell us what this project is? I don't even understand it. Like, who gets paid to do what? I guess would be nice to know.
Well, yeah, that's something we sort of need to figure out to some extent.
Okay.
So, I mean, so the NATO project was really awarded to myself at SFU, Lily Castro, and Jaime at University Industrial Santander in Colombia, the University of Aberdeen with David Dainisi, and Pernilla Jensen at DTU. So we're like the 4 project PIs. And finally, we held the kickoff meeting last week on October 7th. So that was one of the requirements that NATO had of us to actually get the money from them. So we've so far, it's been a long road to get the administrative startup things going, like an IP agreement and a research agreement between the 4 institutions. So that is finally completed and has been sent to NATO in the last couple of weeks. And so that was a lot of work, especially by our legal team at Simon Fraser University, to get that going. So finally that's done. We've had the kickoff meeting, so the project has officially started. And we're still working on getting a few approvals for getting students to be funded by stipends, because there's lots of rules that NATO has about how that works, and they have to approve people before we can start paying them. But yeah, so it looks like right now we're going to have Well, we have involved in the project all four of the project PIs, Lily, Davida, myself, and Pernilla. And in my lab, we have a postdoc who's also going to be doing some of the project management work named Jung Soo, who I will start inviting to these meetings. And then Stella, a master student who's just starting. And then in Columbia, in addition to Lily, of course, there's Jaime Jaimes, who's going to be working on this project, and then another master student named Henry, who hopefully will start coming to these meetings too. Yeah, and so the next steps that we have for the project are basically we need to start doing some revisions to the project schedule and budget already, just because of there were some delays in getting the project started based on like our anticipated notification date, which was later than we thought. So we're going to have to, in order to start the reactors next summer, I need to nail down from Annie when we can actually start those, like what month. We're going to have to do some finagling of the schedule, and then we'll have to sort that out with NATO. But otherwise, the next steps are really like getting all of the data from Annie, having a chat with Annie about figuring out how we organize running these reactors at NREL, or what the alternatives are possibly. And then looking at the design of all the digesters that we already have, looking at together with the data that you guys have collected, in order to inform the design of the next reactors, mainly by, well, it's gonna be led by Davida, as well as Pernilla, and then together with everybody else from the CHAD group that wants to be involved as well. Yeah, so I think that's basically the update I have. If anyone has any questions, I'm happy to try to answer them at least.
Oh boy, I have opinions.
Good.
I mean, I've been looking at small reactors for a long time, and it's fascinating that you're going to do that. So I might chime in and say, well, here's the sort of universe as I know it of different form factors and stuff of small reactors. And yeah, so that'll be great. I'm looking forward to that. You said Jaime. Is Jaime this Marti?
No,
or is
it's
this Jaime
Jimenez.
Jimenez. Okay.
Jaimes, yeah.
Okay.
He works in Columbia. He works with Lily. I think he was actually now has become a professor there as well. But he used to be a postdoc. But yeah, he said
Okay.
he's become a professor too. So great. And I think Jaime Marti Herrero, he's involved in the CHAD group. Hopefully he works with Lily a lot, I think, on the digesters that they run. So hopefully he'll be involved in the design as well.
Sounds good. I love it. I love it. So you had your kickoff meeting, but the tag thing hasn't actually started. That's a separate like group.
Yeah, so basically, I think the first step is we need to get the data and maybe talk to Annie and anyone else at NREL who knows what went on for the last year, get a full update on that, and then start working on the design of the next reactors.
Yeah, so Jane, should we what's your readiness level for scheduling a call and just talking about all that specifically?
I would be ready to talk. This week's not great for me, but after like towards the end of next week or beyond would be a good time. So maybe I'll send you an email. We can just set up a time that works.
Okay, great.
Yeah.
Late next week or after for me, I'll be traveling Monday to Wednesday.
Okay, next Monday or the Monday after?
Next Monday, I'll be traveling.
Yeah, so after Wednesday next week. That sounds good.
I will just say beginning coordination with NREL in coming weeks.
Okay.
That's great. So awesome. Looking forward to hearing more. And what about social factors? Is that part of the research?
So social factors, you mean in terms of like in the communities?
Yeah.
So social factors, I would say, are not really considered. What is being considered is like what kind of waste is available and what kind of materials would be available,
Cool.
both to build the digesters, but also to feed the digesters.
Nice. Cool. All right. Let's wrap it up here. This whole thing about IWA stuff from Angela Bywater, I have no idea what this means. Does anyone want to say anything about the IWA stuff I put in well, it was in the agenda, but it was also in the chat. Scaling biogas globally, WBA Connect and Community Roundtable, 3 to
I
4
think
p.m.
I think
UK.
I can talk
Yeah,
a little bit about that.
chime in. Yes.
There is some enthusiasm from I think Chad members who are not on this call, which is kind of funny, for organizing a group participation in a conference. Embry Bronstad is a big
Ah,
conference attending person. Um, and so she's thinking it would be great to have a sort of in-person meeting and this conference because it's at Aberdeen and we have two members of this consortium in Aberdeen. I think she feels like that's a node that we can build on. It's next year about this time, I think. And maybe IWA is International Water Association. I'm a little vague on the details. I never get travel money. So and I don't understand, I can't predict the future of the US federal government anyway. So it's a lovely idea, and I encourage anyone who's interested to connect with Embry and Tom Curtis and Robin and other folks who are closer to Aberdeen, Scotland, and are more likely to attend. That's what I know about it mostly. Yeah.
Yeah, I
cool.
think I can add a bit more context to that. So it's actually in Glasgow, it's not in Aberdeen, but pretty close to Aberdeen. Scotland's not that big of a country. So Embry, Tom, Robin et al. Are preparing a proposal for a workshop on this topic, and they are hoping to have as many of interested Chad members attend as possible. We do have travel funds in the NATO project to support people who are working within that project. So the students working on that, and I think the PIs as well should be able to attend. And then, we're trying to find, I think, sources for supplementary funding to help other people attend as well. I think what Angie said, though, is something different. That's for this October.
Right.
So there is an event being held online by the World Biogas Association in conjunction with the IWA. I'm not too sure. But it is, there's the meeting online on October 22nd from 3 to 4 p.m. I'm not sure whose time zone that is. About drivers of regional biogas growth. So I think she was just trying to advertise that.
Yeah, now this rings a bell about the possible workshop proposal, which I think, what is this, like next summer or next fall that this
In
event?
October next year in Glasgow, but the call for workshops because it's this massive conference, it's 10,000 people or something. So the call for proposals and abstracts and stuff ends really early just because of all the organizational steps. So I think it's coming up probably in November, the deadline.
Okay. If you want my help on that, let me know. I could just throw a few good words in there. It's always good to try to capture people's imagination somehow. But then again, they also want it to be rigorous. So whatever.
Yeah, I just got a copy of the proposal from Robin this morning. I haven't had time to open it, but I'll
good.
Forward it on to you.
Yeah, sure. Brilliant. Anything else?
I have a quick question. I just put in the chat, Jane, you mentioned Pernille, and we had received, this is forwarded from someone else in NREL. This is an invitation we get to the WASH Winter School. I think maybe it's looked like different things over time, but I was just wondering if you had heard of this, if this is something that I or anyone here has attended, helped develop, or finds relevant.
Kristen, does this ring a bell? Also, yeah, Jane, I don't know, Kristen, you can see this comment. Water sanitation hygiene winter school 2026 in Ujuar Pik Juwa. It's in Nunavik. Let's just say that.
I haven't heard of that before. That doesn't necessarily mean much. It's probably organized by somebody in Quebec, I would guess. So,
Okay.
yeah.
Okay,
Jasmine?
I think I actually have, and I actually was gonna I don't think that this is new, and I was gonna attend this program at some point in my PhD during a summer is this during summer or like is this like a program, do you know?
It's late March, early April.
Yeah,
which
I
means-
think I have. I mean, I do think it's a really great opportunity, especially with the people that are involved. I was gonna go because they have a really good wastewater system in the way that they just are handling not only honey bucket waste, but I'm pretty sure the wastewater facility within itself. So yeah, I think it's a great opportunity. I definitely would look into it if you have anybody that wants to do it. But also I'd like to say that with the new reactors, just as a student, I would love to be involved in any aspect in terms of like I love to learn. So if I could ever be involved just to be sitting there, please let me know.
Yeah, I think that would be great.
I'm really big on opportunities and learning, so I'd love to just know what's going on.
So what is your PhD research about actually? Like integrated somehow into this project?
Yeah, for sure. My so right now, like I'm finishing my master's, and so that's the anaerobic digestion model that I was showing you guys when I presented. I'm finishing that, and then I'll be transitioning within this upcoming year into my PhD. And so the goal behind that is with some of the work that I was reading that Tom Curtis has done, he has gotten some of his digesters to work at 4C, 10C, 15C. I'm interested in those very low temperatures and getting my anaerobic digesters to work. But through talking to him, and I think I mentioned this before, I'm going to be looking into lipolytic organisms, specifically psychrophilic lipolytic organisms. And I want to be a lot of my influent to be really, really rich in fats, oils, and greases, just because.
Right for the hardest things. Yeah.
Exactly. Yeah. I know. I'm just going for the hardest things. So my thing is I'm wanting to, I think we're going to be creating like a population first, culturing a population of lipolytic organisms. And then we'll be inoculating them to see if I can get them to run around 4C, 10, and 15C. But I think we're going to be focusing more on like how well identifying the lipolytic organisms themselves and also their enzymes specifically, because there are a few things that Tom had shared that they had done some research and it didn't really go out the way that they were wanting it to in terms of identifying certain aspects of those organisms. So I'll be basically doing a lot of the work that he's already done and just kind of filling in the gaps where he wasn't able to either continue the work or it's just not of importance as of right now. He was really excited when I brought it to his attention. He was really for it. But yeah, so just inoculating microorganisms to see if they can run at lower temperatures and digest the waste.
So 2 quick questions. So not that you have time, Guy, but a couple months, was it 2 months ago or 1 month ago, Jasmine, you gave a presentation on your model, which was like, it sounded really powerful.
Yeah, I'm looking back through to see if those presentations are on the Chad.
Yeah, there'll be a YouTube, Jasmine. You definitely, yeah.
If it's all right, yeah.
Yeah, I'm
I'd
on.
love to see that.
I love that we record these. It's great. But yeah, what was your question about the model? Were you asking me a question or?
I was just telling Guy to go look for it. Was it 2 months ago
Yeah.
or not?
About 2 months ago. Yeah, I think so now. Either a month or 2 months ago. But if you just type in my name, or I think it'll come up. Yeah.
Okay.
Cool. So yeah,
Thank you.
we'll circle back, Jasmine, to see when, whenever you're ready to tell us more about it. It's pretty wild. I guess the other question is, would the model handle not, something other than psychrophilic, or is it only for psychrophilic?
No. And the anaerobic digestion model 1 was built off of just like a mesophilic anaerobic digester. And so like, of course, over time, people have applied it to thermophilic as well. And so it can be applied to all 3 really. What it comes down to is just really being able to understand the chemical oxygen demand, what you're feeding it, and then how that breaks down numerically, right? So that's just like the core piece. But yeah, it can work for all different types of models. And I'll be trying to, through my PhD, trying to get it to adapt it to a psychrophilic anaerobic digester, but that's going to take some time.
So it sounds pretty similar to some of the work I think that Lily and Jaime are hoping to work on in in the NATO project as well, like trying to like adapt inocula to lower temperatures. So it would be nice for you guys to interact a lot on that.
Yeah, for sure. It definitely would. That's awesome. That's exciting. So exciting.
Margaret asked about if there's a link to this WASH event. I also want to share that with Judith, who already may know about it. Marachel. She's pretty active up in that area. So that might be the way to find out, Margaret. I'll tell you what, I will ping her and copy you, and then we can get it out more broadly when we have a link. Okay.
Yeah, I guess another plug I could put in here. So I'm involved in it's a training network for graduate students in Canada called WASH Canada. And we're not linked up with these people organizing this, but we do hold WASH Canada talks. I think it's every Thursday morning at 9 a.m. And we get like a lot of really interesting people, you know, people from the UN, people from the WHO giving talks, and then a lot of professors kind of from all over the place that work in the WASH area. But I might just I'll put the link for that in here so you guys are aware of that.
That could be a feeder network for potential acolytes.
Oh, it definitely is, yeah.
Yeah,
and
so
Stella, the master's student who's going to be working on the NATO project, is also part of this WASH Canada program.
yeah,
too.
cool.
And that's led by, I think you guys have met Caetano Doria. He's a professor at University of Victoria, and one of his students gave a talk. I think she gave a talk here a few weeks ago. Kelsey Shaw, maybe.
I don't think we had that person, but maybe we should.
Maybe I'm thinking of another connection. Okay, well, anyways, he knows about this group, and you know, is interested in theory. One of his students, I think she's a postdoc now, Kelsey, is working on sort of more on like what's happening in septic systems in cold environments. Sort of similar to anaerobic digestion, but in a slightly different context.
Great. Thanks for all those links, everybody. That's great. So many great connections to be made. Thank you. All right. Well, we went the full, whatever, hour and a half. Wow. Okay. Thanks, everyone. Anything else?
Can I keep coming back?
That's another thing they say in 12-step meetings too. Keep coming back. Yeah.
Yes,
man,
please do keep coming back.
Yeah.
If you can figure out
you.
how to be
Yeah,
part of
really interesting.
IRPIC, then you qualify. If you get caught in password hell, then, you know, sorry, buddy.
No, if you get stuck in password hell, you just contact Meredith, because that's what the beautiful thing about having the IRPIC
Yes.
digital home is that it has support, and
Yes.
we can the document storage is still a little janky, but I'm sure that'll get better over time too. And yeah, it's supposed to be a welcoming digital home, despite what Alex says.
Oh, thank you, Meredith. That's great. I'm really curious. Yeah.
Okay.
Hear more.
Well, you're not any help because you already are a proselytizer. So, you know, we're looking for new recruits, like
Right.
Wilder. We want to cultivate them.
Brother. Do we have access to that presentation? Would you mind sharing? I would like to show my lab group those pictures of your reactor that you built, if that was okay.
Yeah, absolutely. Yeah. Let me send this on to as long as you recognize that this relative proportion is just hypothesis, a lot of unsubstantiated discussion there. So just consider them hypotheses. Great. Yeah, I'll forward that to Alex, and maybe that will find its way.
Yes.
Or should I send that to Meredith, or what's the best way to do that?
Margaret, if we can do a document storage version of it, let's do that. But
yeah, we can. I think pretty much anyone in the group can upload things to the community of practice site. I mean,
Okay.
you do have to be a member, and then you can upload documents. The thing that's particularly janky is that it's not like there's a sort of good viewing window for that. There's a search tool that works, but you have to know what you're searching for.
Okay.
So
Sounds good. Why don't you do this, Guy? Send it to me and Margaret. I will not only upload it, but I will send it out to Jasmine.
Super. Thank you, Alex.
And actually kind of practice that thing myself of what Margaret's saying. All right, last thing. Anne E. Corrigan, Margaret McCauley, welcome back.
still here. That's
Still
that's
here.
all I can say.
I think that was it. Yeah. I'm not a federal employee, if that
Oh.
was what that shout out was for.
Kristen
Fortunately,
is.
I am just a contractor.
Oh,
but
okay.
yeah,
Well,
Margaret,
welcome back,
Kristen,
Kristen.
I appreciate
Yeah,
seeing you
thank
here
you.
today.
Yes, I'm also a federal employee and I'm still here.
Yeah.
Good luck.
Yep.
I don't know if I'm getting paid, but I'm here. So that's current status.
Yeah.
All right. Thank you all. Lovely to
Thanks.
see you.
Good to
Yep,
see you all
same
today.
here.
On
More
that note,
soon.
See you next
Good one.
cheers. Thank you so much. Thanks, Alex.
You bet. Over and out.
Really fun. Cheers.
Yeah.
Bye.
Good. I thought you would think so. Bye.
Yeah, it's really cool. Cheers. If I can figure out how to turn off.